Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this session to discuss NCERT Class 12 Chapter 4 Principles of Inheritance and Variations. Part 6 Chromosomal Theory of Inheritance. The Mendel published his works on inheritance characters in 1865. So Mendel he did as an experiments in 1865. But several reasons the remained unrecognized till 1900. So up to here 1900, here his characters are not they are published. What were the reasons? See that in a reasons one by one. The first reason. Here the first reason. See the first one, the communication was not easy, and it is now those days, and his works could not be there widely published. So in the time the communication like nowadays we are seeing that in the Twitter, Facebook, Instagram, so whatever this online communication is very well developed, but this kind of communication those days are not there. So that purpose his complete communication is not proper at that time. Okay, that is the first one. That is in a first one. And second one is what? His concept genes or factors in the Mendel would that their stable distinct units controlled by the expression of the traits and the pair of alleles did not blend each another. It was not accepted by the complementaries and in the expansion of their apparently continuous variations in the nature. So second one is what? So Mendel is saying about as there the two contracting units as present. Yes, now these contracting units does not they're blending the F1 generation. So already you crossing the two characters, you crossing with as in these two characters already getting as in the F1 generation. Yes, now so he is saying about this not blending, but other side is not accepted that one. So that is in a second thing. Next third one. What is third one? The Mendel is the approach as using as a mathematical explanation biological phenomena that totally new and unacceptable to the many of the biologists in this time. So here the third one is what? So first time the mathematical problems and statistical analysis their first the Mendel used as in the biology. So other scientists that is not accepted. Okay. Next last finally the Mendel work suggested that as in factors the genes distinct units could not they provide as any physical proof. So fourth one is what? Finally he not given as any physical proof. Okay, the extents as in a factor say which as in that made up of. So which factors they are made up of he not given as in a physical proof. So these factors based on the four factors are there. So that purpose his characters are not well distributed as any entire in that academic years. So after in 1900 the three scientists De Vries, Corncorens and the Sharma the independently the rediscovers the Mendelian principles. So Mendel principles they are rediscovered and inheritance of the characters. Also by in their time the that advanced microscopy also that takes place. So at the time what happened? The advanced microscope also they are available. So that microscope also available. So the number of the how to the cell divisions was takes place. What is the chromosome structure? How do the chromosome get separated? The entire everything is clearly there explained about as in the different scientists. The scientists were able to careful observation of the cell division. See here how to the cell will be they divided. How do they get cell to be they are divided and leads to the discovery of the structure of the nucleus and appear to as a double standard and double and divide just before of the cell division. So before the cell division what happened? After the cell division what happened? They clearly they are noticed. These we are called as any chromosomes. That is in color bodies. We are visualized the staining. So when the staining what happened? They clearly they are visible. Okay. By in 1902 the chromosomes movement during the meiosis has worked out. So in the 1902 very important this word. See it is in 1902 very important this word. In 1902 the chromosomal movement during the meiosis. The G1 phase it is in the chromosome. It is what? It is in a chromosome in that yellow body and red and is in orange and green. So in that after G1 phase to the G2 phase before what happened the S phase the complete the chromosome here what happened the duplication is takes place. In that S phase what happened here the chromosome, their replicate stage takes place. So they get formed as what? The bivalent chromosomes are formed. To get formed as what? Here one to get becomes as in a the two chromosomes. Here to get formed as in a two chromosomes. So in that, when you observe to as in that meiosis 1 of anaphase, what happened? The chromosomal, the segregation means that anaphase, in that anaphase what happened? The total, the two acids to get their pull up, then what happened? To get forms as in a, the chromosome separation will be takes place. When that chromosomes they get separate will be takes place, then after in that process of the telophase, what happened? They are getting as in a, the four their germ cells. They give as in a, the four the germ cells are nothing but as in a gametes are formed in that process. So they clearly they observed as in a their scientists on the electron microscope. Okay, and see they give the meiosis and the germ cell, the formation of the cell which form the four chromosomes. You see in that in a chromosome segregation means the germ cell after they are formed. Okay, let us see here what is the comparison of it there between their chromosome and gene. What is the comparison? See here this is a chromosome. It is in a the chromosome and it is what? It is a gene. It is what? Gene. Chromosome and then gene. See, it is occurring pair. The chromosome it is occurring as in a pair. Okay, chromosome occurring pair. It is also there completely occurring as in a pair. Next, the segregation of the time of the gamete formation such as the only one of each pair that transmitted as in a gene. So here to there some they're transferred as in that parent to as in a gamete. Here, parent to gamete. They transmit as in what? Some genes. Here some information to get they get completely get separated. Means segregated. Here also the same. The segregation of the gamete formation only one pair. Here there one gamete. Here one pair they transmitted into as in a gene. Next, independent pair of segregate independently of each other. They are independently they get segregation. And here one pair of the completely the segregate independent of any each another. Here also they completely the pairs also they get independently they get separated. You can tell which of these the column A, column B the represent that in a chromosomes while they represent as a gene. How did you decide it? So most of these are both are completely they get act as what the genes that both are they having as the genetic information. Okay, like let us see that the phase of the during the anaphase the meiosis 1. Here the completely during the anaphase of meiosis 1 the two chromosomes pairs can algin at that in metaphase plate the independently of each other. The chromosomes are they completely there independently of the each other. Next, understand this the compare the chromosome the four different colors the left and right columns. So here they given as in that four chromosomes are they given the four chromosomes are given as in that left column as well as in a right column. The left column the possibility orange and green. See orange and green. See these are the completely orange and green and here the left column orange and green together but the right hand the column the possibility that orange chromosomes. So when they get completely they get segregation will be takes place. What happened in the right hand column the possibility they given as in a the 2 in that possibility 2 they given as what here orange and then red column they given. See here orange and they get red chromosome they are given in that possibility 2. It is a possibility 1 it is in a possibility 2 is they given. And see in that possibility 1 they given clearly here. The first we see that here in that process the long orange and short green chromosome, long yellow and short chromosome at the same pole. One is in a long orange and short green chromosome and one is in what long yellow and then here they're completely short red chromosomes are present in the possibility 1 that is the meiosis 1 anaphase. Here possibility is there 2 that is in a long orange and short red, long yellow and then short green here they given. So when they get after crossing, see they get forms as what? Let us see the germ cells also they get producing the same. Here they get produced as in a that same the germ cells are produced. The independent assortment of chromosomes. So independently they get separated. When the anaphase only they saying that one only. The two acids they get pull up, the chromosomes the separation will be they get takes place. So independently the chromosomes are formed. Okay, that is. Now let us. After the Sutton and Bovary argued that is a pairing of the separation of the chromosomes would be the lead the segregation of the pairs the factors is carried. So here there scientists Sutton and Boveri, they argued with as the chromosomes, they separate the segregation, the pair, it is some factors here, the information they carrying from as one gene to another gene, how this takes place. Here some their carriers as present, here they having some carriers. See, certain united knowledge, the chromosomal segregation with as a Mendelian principle is called as a chromosomal theory of inheritance. So like as in here the certain knowledge, the chromosomal segregation, here the chromosomal segregation within a Mendelian principle is called as what? That is in a chromosomal theory of inheritance. Okay. After, see here, is following the synthesis of an idea experimental, the verification of the chromosomal theory, the Thomas Hunt Morgan. Here the scientist was Thomas Hunt Morgan and his colleagues. So he leads to discovering as the basis of the variation of the sexual reproduction produced. So Thomas Hunt Morgan did as in some experiments on the here some lab experiment he took as one fruit fly. Okay, here he worked as in the tiny fruit fly, that is in Drosophila melanogaster. So he took as did as in some experiments on the tiny fruit fly, that is name is what? They are Drosophila melanogaster, which found as in the suitable such as in studies is could grow on the simple synthetic medium in laboratory. So he did as in that experiments in where very important point. These are it is in he grow as in a complete synthetic medium in the laboratory. There complete the life cycle about as in 2 weeks. Complete their life cycle how many weeks? Within the 2 weeks. The simple mating could be producing a large number of progeny flies. It is also as in that clear the differentiation of the sex, the male and female, the flies easily the differentiable. So here once you observe here clearly, here easy to get notice. Here easy to get notice. Here completely one is the male and one is the female. It is a male is in a small one and the female is in a large one. You can easily to get identified. Means here the sex organs they can easily to get determined. Sex organs easily to get they are determined. Next, after here he also there are many types of heredity variations they can seen within a low compound power of microscope. So that what type of offsprings we are getting, they can easily to get observed in a low compound microscope. Okay, that is next. Remaining to discuss in next class. Thank you for watching. Like, share and subscribe.